Hello, this is Mrs. Kant. I am coming to you with the Unit 2 test review. This is going to go over a topical um, reasoning for what's going to be on the test. It's not specific questions that are going to be on the test, but they are topics that will be on the test. As you go through this, you should be able to study this information, study these topics, and then complete the unit 2.16 unit assessment, which will be found in USA Test Prep, and hopefully be able to score above a 70% on. Let's go ahead and get started. The first question is defining that population, gene pool, and species. Make sure that you get this information down. Population, same species, shared space, be able to reproduce together. The gene pool is going to be all the traits in a population, all of those genes. Eye color is going to have a specific, be a gene. I'm also going to have all those traits available. Brown, green, blue, all of those traits are available in that gene pool. And a species, to define a species, they need to be able to interbreed together as well as produce fertile offspring and be genetically similar. What are those three variations that can occur in a population? That'd be mutation, gene flow, and genetic recombination. How would you describe those? Mutation is going to be a spontaneous change in the organism's DNA. Gene flow is going to be new genes coming in or going out of the population. And then genetic recombination is going to be those genes being able to cross over during meiosis from one chromosome to another. What is natural selection? This is when the natural environment is going to act on individuals for those favorable traits to survive and reproduce. Those that are not favorable will be eliminated. How does natural selection act on a population? Well, it's basically choosing its favorites. It's going to favor the trait that helps that organism survive. Different examples, the white rabbit in the Antarctica, that color is going to help them. Striped snails versus brown snails on those rocks. We talked about that in one of our sessions. Your lab that we talked about, lizard leg length, that is also an example of natural selection. What can we learn about those birds and the different shapes and styles of beaks? If you remember from Darwin's finches, he proposed that the beaks adapted for different feeding habitats or different feeding niches. Two examples of natural selection working on a population. Things like predator preference. Predators maybe like those green feathers, so they're going to go after those green feathers so much better than if they were red or blue. The white moths, peppered moths, the black ones were able to blend into that environment while the predator was eating the white moths. Mate selection is another one. Different color skins on salmon are going to be preferred. Better display of feathers on those peacocks going to be preferred as well. Bright colors. Drawing a graph of moderate selection, be able to explain what it means. Moderate would be that normal, where the highest numbers are going to be in the middle of the graph, and then the lower numbers are going to be on the outside of the graphs. Directional, I want you to make sure that you know a bunch of examples for this one. Directional, where we are going to be tending to favor one extreme over another. So bees, butterflies, birds, if we are eliminating some of those pollinators, what trait is going to be left over in that flower gene pool? Your lab on lizard populations is also an example of directional selection. Natural selection chose the longer leg length on those lizards than the shorter. Disruptive selection, this is where you are going to get that two humps in that graph. Natural selection is going to favor both extremes. So in this example, I have a gray rabbit and a gray and white one. Those populations were selected over the white one, which that population dropped. And then the stabilizing is where that average is going to be chosen over the extremes. The extremes are cut out, and you are going to get mostly that average. Things like a predator moving into an area is going to probably select over specific ones. Babies' birth weights, you have a nice average that is a good for baby birth weight. Those very extreme of really, really low birth weight and really, really high birth weight are not necessarily cut down, but they are very, very low in numbers. Evolution. This is where there's the change in allele frequencies or that genetic makeup. And it can change in any direction, and they're usually small changes over a long period of time. So it's not evolution at in a blink of an eye. It's going to be over many years, hundreds, even thousands of years. How might a predator cause evolution in a population of a prey? Potentially, if we are trying to evade a predator, those prey are going to develop certain traits that help them evade those predators. Explain how scientists have used embryonic development to support the theory of evolution. That would be these embryos. They're comparing different stages of development and the similarities between those stages. That is called comparative embryology. What are those homologous structures? Homologous structures are going to be those that are in different species but are similar structure and arrangement of bones. And they suggest a common ancestor. What are those vestigial structures? Those are the structures that have lost function. At one point in the past may have been important. However, right now they have no use, like the femur and pelvis bone in the whale. Again, is used as evidence for that common ancestor. Homologous vestigial structures. And then the amino acid sequences providing evidence for evolution, those are going to be comparing those DNA or amino acids together between two organisms, looking for those differences. The closer in relationship, you're going to see less differences. More differences are going to be further apart in relationship. Here's that example of organism 100. We look at this, it says number of differences. I have 8 differences here, but only 1 difference here, means that D may be closer related than A, B, and C are. Two reasons fossils are used to provide evidence. They're going to be giving information to that time period that the organism lived in. They also use some fossils as showing transitional forms between different past life forms and how they have changed, those intermediate stages. So can also be used for that chronological order of life. Radiometric dating, what does that tell us? That's going to be determining the absolute age of years in geological samples using radioactive isotopes, tells us a sample age of that rock or that specific organism that's found in that rock. What does it mean to be geographically isolated? Populations are going to be separated by geographic barriers like mountains, islands, rivers. So the separation of populations allows development of new traits and changes among those populations. Here's an example. Isolation of a species will adapt to eat differently because of the food that's available. Certain shells of snails could be different shapes. Skin or fur could be different colors. All females select a certain trait in a male. What would happen to those males in the population? Well, you're going to see a shift in those traits to those more preferred traits by the females. So different mechanisms of natural selection, we kind of talked about this already. Mutation is going to be causing variation. Competition for mate is going to be another one. One allele is selected over another because of that competition between mates. And then that reproduction, being able to pass on those alleles. What is the Hardy-Weinberg principle? Write the definition, formula, and what each part of the formula means. Well, here is the formula, the definition. It's an equation that is used to predict allele and genetic frequencies in the population. What does P mean? P is the more frequent allele, which would be more that homozygous dominant. Q is that less frequent, that's going to be the recessive gene, and then 2PQ is going to be heterozygous. If P and Q are not changing, they are at equilibrium. That means change is not happening and evolution is not taking place. What does it mean when a population is in Hardy-Weinberg equilibrium? It means the population is at a constant allele frequency. No change is happening. How can the gene frequency change? Give an example of how gene frequencies can change. Under natural selection pressures, new predators, mate selection, that genetic drift. In smaller populations, reduced genetic variation, things like new colonies of low genetic variation. There's a random change in the gene pool can have large effects, like disease coming into one of those populations. What is a lethal gene? It is gene combinations that cause an organism to die before it is born. And how do lethal genes stay in a population? Heterozygous carriers, those that carry the gene, so they are able to pass on the gene. However, that means that they are not actually showing the gene. So carriers are going to reproduce, pass on that recessive gene to heterozygous or homozygous recessive offspring. Hopefully you got all that information. If I went too fast, make sure you review this and try looking at it again. You can pause the video at any time, but you should now have all the answers to that review sheet, be able to turn that in for extra credit, and then go and take that test and score hopefully a 70% or above. If you have any questions, please let me know. Have a great rest of your day.